Hi students, how are you? I hope each one among you are doing great and well. Welcome to Supnit's classes. This is Rahmat Latif, your biology educator. In this video, we are going to learn male reproductive structure in detail and how the male gametes are formed and in which structure they are formed. So without wasting any time, let's get started. First of all, I already mentioned, I'm mentioning again, I'll just draw structure. Okay, this is filament, this is bilobed anther. This is filament, this is bilobed structure anther. It's having two lobes. How? This is the first lobe, this is the first lobe, and this is the another lobe. Okay, that means anther is bilobed. And the male reproductive structure is known as androecium, in which we can say it contains, so many stamens. It contains so many stamens. Okay. I hope this concept of male reproductive structure is clear. Okay. This is anther. This is filament. This is androecium. Male reproductive structure is known as androecium. And how this is attached to the flower? This is attached to the flower through which structure? Through thalamus. Okay, I hope this concept is clear. I hope this concept is crystal clear. Okay, now we will see. Okay, androecium means male reproductive structure and gynoecium means female reproductive structure. I am clearing it. Please remember this. Anther is bilobed. It is attached to long stalk known as filament, and how full stamen, it's anther and filament together is known as stamen. How stamen is attached with flower? It's attached through which structure known as thalamus. Okay, I hope this concept is crystal clear. Now we'll see, and I already mentioned androecium means male reproductive structure. Androecium is male reproductive structure, right? Then what's the female reproductive structure known as? Female reproductive structure is known as gynoecium. Please remember. Male reproductive structure is known as androecium. I'll write it down. Male reproductive structure is known as androecium, and female reproductive structure is known as gynoecium. Okay. Androecium consists lots of stamens. Okay, which itself is consisted of stamen and filament, sorry, anther and filament. Anther is bilobed, and gynoecium consists so many pistils, or another name of pistils is carpels. Okay, I hope this concept is clear. And pistil itself is formed by stigma, style, and ovary. This is stigma, the structure following stigma is style, and this bulged portion is ovary, and inside ovary is ovule. And I already mentioned, after fertilization, ovary will get converted into fruits and ovule will get converted into seeds. Okay, I'll erase it. I hope this concept is clear. Okay, now we will see. Okay, we are done with first slide. See, say for example, I am just cutting anther. This is anther, bilobed structure. This is filament attached to it. I'm just cutting this anther in transverse section. Okay, it will look like this. See how it will look like. It will look like this. It will look like this. Okay, right? Very nice. This is this portion is one lobe and this portion is another lobe. That means it's having two lobes. Okay. Anther is bilobed. This concept is clear. Anther is bilobed, right? I show you the upper portion is one lobe, lower portion is another lobe. One lobe, second another lobe. Okay. And in each lobe, you will see that it's having two theca. It's having two structures like this, right? One and two. In one lobe, it's having two theca, and in another lobe, it's having two theca. That means we can say each lobe is dithecous. I'll write it down. Each lobe is dithecous. I'll again make it. I hope this concept is clear. Now I will write it down. What is, bilobed? What do we mean by bilobed? And what is dithecous? Okay, I'm drawing it again. Like this, like this, like this, and this. And above the portion which is above is known as is one lobe, and the portion which is downwards is another lobe. And each lobe is having two theca. One, two, downwards 1 and 2. It means each lobe is dithecous. First of all, please remember anther is bilobed. This should be your first point about anther. Anther is bilobed, and then you can write each lobe is dithecous. That means in total we have four. In one lobe we have one theca, another theca. Another lobe we have one theca and another theca. Okay, that means in total we have four theca. That is tetrathecous. But if we will talk about single lobe, it is dithecous. Okay. I hope this concept is clear. Your point should be that each anther is bilobed, and each lobe is having two theca. That is, they are dithecous. In general, anther is tetrathecous and bilobed. Okay. Inside each theca, we have structures like this one, which appears to be, circular, 2, 3, and 4. These structures are, they appear to be circular in shape. These structures are known as microsporangium. Okay. Inside each microsporangium, we have tissue, and the tissue is known as sporogenous tissue. Sporogenous tissue. Since this is tissue, that means they are formed by some cells, and those cells are known as MMC or PMC. MMC means micro mother cell and PMC means pollen mother cell. Okay. And after, maturation, these MMC microspore mother cell will get converted into, pollen grains. I'll show you how. Okay. First of all, you please remember this procedure. What MMC is and what PMC is. Okay. They are, they together form, these cells together form sporogenous tissue, which are stored inside microsporangium. Okay, since these are very important cells for us, they are going to get converted into pollen grains, means male gametophytes which contain males. They are very important for us. Okay, that's why we just cover them through covering. First covering, first covering of this microsporangium is known as tapetum. First covering is known as tapetum. I'll show you one and the other four will be clear, because same would happen with these, all these. Okay. These layers are known as tapetum. Tapetum, and tapetum contains which cells? Tapetum contains tapetal cells. Tapetal cells, they provide nourishment. Their function is to provide nourishment. They provide nourishment to growing microspore mother cells or pollen grains. Okay. And that's why these tapetal cells are binucleated. They have two nucleus and they have dense cytoplasm, because they have to provide nourishment. Okay. And the outermost layer of this, microsporangium, the structure is known as epidermis. Okay. Epidermis. And after epidermis, we have one more layer known as endodermis. Then we have middle layers, and after middle layers we have tapetum. Okay, I hope this concept is clear. And in between here, we have some tissue known as connective tissue, which connects all these theca and lobes. Okay, I will write down the. I hope this concept is clear. I am repeating. We have in each theca we have microsporangium, and microsporangium on its inside contains sporogenous tissue, which itself is formed by microspore mother cells or pollen mother cells. And microsporangium is very important for us. That's why we need to protect this. Just like if we are protecting gold or money in our home, similarly we have to protect this because they are going to be very useful for us. Okay, that's why we have just four coverings of we have just four coverings of microsporangium. I'll write it down, but you will remember microsporangium appears circular, okay, in transverse section. let me write down the Let me write down the name of the layers. Okay, please revise it continuously. Do make notes so that you won't get bored, or it won't get burden at the time of examination. See, micro we have four layers of microsporangium. First layer on its outside is known as epidermis. It's outside layer. Okay, outermost layer, epidermis. Then we have another layer followed by epidermis, it's endodermis. Then we have some layers known as middle layers, and finally we have one layer known as tapetum. Okay, epidermis is outermost layer, and tapetum is the innermost layer. Okay, what's the function of innermost layer? That is tapetum. It provides nourishment to the growing pollen grains. And what's the function of epidermis, outermost layer? Its function is just to protection. Middle layers, endodermis and epidermis just provide protection. Okay, I'll show you what will happen. We will just bring out the one cell of sporogenous tissue, that is microspore mother cell or pollen mother cell. Each microspore mother cell or pollen mother cell, they have the ability to grow into what we call it as, you know, to give rise to pollen grains, which further gets converted into male gametes then, okay? First of all, listen, pollen grain is not a male gamete. Please remember, don't get into this trap. Don't get into this trap. Most of the students are confused. They are saying that pollen grains are male gametophytes. Sorry, they are saying pollen grains are male gametes. No, they are male gametophytes. Pollen grains are male gametophytes. Male gametophytes means which carries male gametes. Which carries male gametes. Inside pollen grain, the pollens which you see around you, okay, which gets travel through air or some insects, okay, which we learn in pollination, they are nothing. They are male gametophytes. They are not male gametes. Please remember, they are male gametophytes. They carry male gametes. They are not male. They are not themselves male gametes. They just carry male gametes. That's why they are known as male gametophytes. Okay, and these pollen grains gets mmc, which is microspore mother cell. They will get converted into pollen grains. Okay, microspore mother cell will get converted into male gametophytes. See what will happen. We will bring out one mmc, microspore mother cell, or PMC, another name, pollen mother cell. Okay, its polarity is 2n. It is diploid because it's a part of plant. It's a diploid. What will happen to this? What will happen to mmc? It will undergo meiosis. it will undergo meiosis first. Meiosis of two types, meiosis 1 and meiosis 2. If you don't know what meiosis is, we are going to learn this in class 11th chapter, cell division. But for now, please remember, meiosis has two parts, meiosis 1 and meiosis 2. Meiosis 1 is reductional division. The polarity gets half. Okay, reductional division, polarity gets half. It's a cell division. It will divide the cell. MMC is like this. It will divide this cell into two. It will form like this, 1 and 2. But the polarity is not 2n, because meiosis 1 took place, reductional division. This is n, this is n. Okay, after meiosis 1, meiosis 2 will happen. And meiosis 2 is just like mitosis, means equational division. Division will happen. This one will get converted into two. Okay, this one cell will get converted into two cells, and it's equational division. That means polarity won't get half. It's not n by 2, it will be same, n n n n. And these these are connected through a connective tissue like this. Okay, don't get confused, don't relate this with anther. This is just this just forms microspore tetrad. There are four. Microspore mother cell will give rise to microspores. Microspore mother cell, like mother gives birth to baby. Similarly, microspore mother cell will give rise to microspores. And since they are four in number, they are known as microspore tetrads. Okay, microspore tetrads. I hope this concept of microspore tetrads is clear, right? Microspore tetrads is clear. Now we will took this one, this microspore tetrad, okay? We will took this microspore tetrad, we will see what will happen to this, okay? Hold on. Okay, say for example, I am taking one microspore like this. I have this, I have this microspore now, whose polarity I already mentioned is N. Each now we got from one microspore mother cell, we got four microspores, which is known as microspore tetrad. Each microspore will develop like this. Microspore will get converted into pollen grain. This microspore will get converted into pollen grain. How? What would happen to this? Microspore will get will will gets under mitosis. Let me show you. It What will happen? Mitosis will happen. Microspore will get under mitosis, will goes under mitosis. Then what will happen? Mitosis. Mitosis is also equational division. Mitosis is also equational division. Please remember, mitosis is equational division. That means this n will get converted into 2n, but they are not, they are not separate from each other. They will be like this in one cell. One cell, another cell. That means one cell contains two cells. Okay, this larger and this smaller. This larger cell is known as vegetative cell, and this small cell is known as generative cell. Vegetative cell just provides nourishment to generative cell, because generative cell will undergo another mitosis. Then what will happen? Generative cell will get converted into two male gametes. Okay, generative cell will get converted into two male gametes. That means each pollen grain contains two male gametes, but here we have condition. That condition I'll discuss in our upcoming session. But here remember, microspore will undergo one mitosis. What will happen? It will get converted into vegetative cell and generative cell. Vegetative cell, function of vegetative cell is just to provide nourishment. Okay, its function is just to provide nourishment to generative cell, and generative cell will undergo male gametes. It will get under division, cell division, and it will give male gametes. Okay, I hope this concept is clear, right? Now we have one more concept of this, how this pollen grain will form two male gametes. Now you have to remember, pollen grain is not male gamete, pollen grain is male gametophyte. That means pollen grain contains male gametes, but it's not male gamete. It itself is not male gamete. Okay, please remember this concept. I hope this concept is clear. I'll try to revise this concept in our upcoming session, but please make notes of this, make this flow chart on your notebooks. Okay, I'll meet you in such any other session. Till then, goodbye, take care. Thank you.